Good evening, all. Hope you all can hear me. With the permission of Principal Sir, I will start the program right now with 7. So, welcome you all again to this international webinar on comprehensive current approaches to oncogenetics and pollination ecology. I am very much glad to welcome both the speakers. Priyadarshini is already here and Dr. Sayan Chakraborty is already with us. And I would like to welcome all other participants, Principal Sir, the coordinator of our college IQAC team, Dr. Abhijit. So all the participants are hereby requested. I just want to humbly request all the participants to mute their microphone and to switch off their videos for the hassle-free streaming of this particular international webinar. And now I would like to request Dr. Sumana Shaha, our head of the Department of Zoology, Barasat Government College, to say a few words to the audience.
Good evening, everybody. It is a moment of extreme pleasure to welcome the guests who are present here to attend this webinar. On behalf of the Postgraduate Department of Zoology, Barasat Government College, I offer my regards to our Principal Sir, Dr. Samar Chattopadhyay, TC Secretary, Dr. Arun Roy, IQAC Coordinator, Dr. Abhijit, all the senior teachers, faculty members, my colleagues, friends, and my beloved UG and PG students joining us. I convey my regards to the young speakers of the day, Dr. Sayan Chakraborty, Research Assistant Professor and NMRC Young Investigator, Institute of Molecular and Cell Biology, Singapore, and Dr. Priyadarshini Chakraborty Basu, Assistant Professor, Mississippi State University, USA. Thank you so much, Sayan and Priyadarshini, for accepting our invitation and coming over for the webinar. Wish you good luck for your bright career. The schedule of this seminar will focus on the mechanobiology of Agrin in cancer and tissue healing by Dr. Sayan, and transdisciplinary approaches to improving pollinator health by Dr. Priyadarshini under the broad spectrum, comprehensive current approaches to oncogenetics and pollination ecology. I wish this webinar enlightens us all and provides us with approaches to deal with the problems of tomorrow. Thank you to everyone who made this webinar possible with their opinions and views. To conclude, I wish you all great success in your pursuits. I request our Honorable Principal Sir to address the gathering virtually by his inaugural speech. Over to Principal Sir, Dr. Samar Chattopadhyay. Please, sir.
Thank you, Professor Shubhana Saha, Postgraduate Department of Zoology, Barasat Government College. So, I understand very little about zoology, but what I apprehend from the heading is that this webinar is something, a strategical mechanism about controlling. First one is the controlling cell thermodynamics and cell entropy by some geological tools like Agrin and to control cancers and tissues. So it is basically, I apprehend, is controlling cell thermodynamics and cell entropy in a macroscopic level, and also a strategical modern approach to address the problem. And second one is also some strategical, but it is some improving mechanism of improving the pollinator health by transdisciplinary approaches. So through these two modern approaches, I think this webinar is going to be a grand success, and I thank all the teachers of the zoology department to arrange such kind of webinar of international standard, and will be addressed by scholars of present era from abroad, Professor Sayan Chakraborty and Professor Priyadarshini Chakraborty Basu. So, I think we'll enrich much about this from this webinar, and I thank all for participating in this webinar and welcome all the teachers from different colleges, universities, and particularly the students of different colleges and universities who are attending this webinar. They will gain some insights and learn about what is happening in modern trends of a macroscopic subject, which is also a controllable one through these strategical approaches. So I also thank the coordinator, IQC coordinator of Barasat Government College, who is also present here and will also deliver his address. So again, I thank the speakers in advance and everybody. So with this, I end my speech here. Thank you all.
Thank you, Principal Sir, for the nice introduction, and also our departmental head, Dr. Shubhana Saha. Now, I would like to request Dr. Abhijit De, Associate Professor, Department of Physics, and the coordinator of our IQAC cell, to deliver his speech. Sir.
Well, good evening, everybody. On behalf of the IQAC Barasat Government College, it is my pleasure to welcome Dr. Sayan Chakraborty and Dr. Priyadarshini Chakraborty Basu for delivering us a very interesting. I hope it will be truly interesting, transdisciplinary. I mean, as I understand, it is rather an interdisciplinary area. And I also understand that actually sky is the limit, because the phase which we are now going through is truly a different kind of teaching for us. And I think that if we are only classically sticking to our own subjects, it is rather impossible to tackle all the problems presently we are facing. Actually, we have been losing many good friends, relatives, and that is really a negative impact on us. But anyway, we have to again fight back. I only hope that this kind of platform and the webinar will give us the true impetus to go ahead. So I welcome both the speakers on behalf once again, and I really appreciate our principal to go ahead with this kind of inertia in the true spirit. So I welcome you all again without taking much of your time. Thank you very much.
Thank you, sir. Thank you very much. So now we are going to the main part of our webinar, and I would like to request Mr. Chakraborty, one of our MSc Semester 3 students, to introduce our first speaker, Dr. Sayan Chakraborty. Dr. Sayan Chakraborty is now actually serving as a research assistant professor and NMRC Young Investigator, Institute of Molecular and Cell Biology, Esther, Singapore. So, Pritam, start.
Thank you, sir. Good evening, everyone. Thank you for joining us for today's webinar. Myself, Pritam Chakraborty. I am honored to get the opportunity to introduce today's first speaker, Dr. Sayan Chakraborty, Research Assistant Professor and NMRC Young Investigator, Institute of Molecular and Cell Biology, Singapore, who will present the machinery of Argin cancer and tissue healing. Dr. Chakraborty grew up in Kolkata, India, and has a master's degree, first class, first rank in biochemistry from the University of Calcutta. He did his Ph.D. in microbiology and immunology from Chicago Medical School, Rosalind Franklin University of Medicine and Sciences, the USA, where he worked on the biology and entry mechanisms of oncogenic human herpes virus 8, HHV8. In his PhD work, he showed that HHV8 utilized Agrin A2 receptors as a master signaling assembly of establishing infection and oncogenesis. During his postdoctoral work, his studies provided the first evidence on the role of argin in mediating liver tumorigenesis. Currently, he is a senior research assistant professor at the Institute of Molecular and Cell Biology, ASTAR. His research is focused on the elucidating mechanisms of argin-mediated oncogenesis, devising strategies to target argin and liver cancer, and exploiting argin protein therapy for effective wound healing. He has published several seminal discoveries on leading research journals, including PNAS, Nature Communications, Cell Reports, PLOS, Pathogens, Oncogene, and ACS Chemical Biology. Dr. Chakraborty is the recipient of prestigious NMRC Young Investigator grant in Singapore, equivalent of K99 in USA, and serves as PI and co-PI for editorial board, co-PI for several competitive extramural grants focusing on cancer and tissue healing. Besides, he also serves as an editorial board member of Translational Oncogeny Elsevier, Cancer's International Journal of Molecular Sciences, IJMS, and Frontiers in Molecular Biosciences. Now, without any further ado, we will turn the time over to you, sir.
Well, good evening all. Thank you, Pritam. I hope I'm audible.
Yes, sir. Yes, sir, you're audible.
Okay. So it's a great pleasure to be here in Barasat Government College, and I really thank the opportunity to be presenting my research here. First of all, I'll thank Drs. Chakraborty, Dr. Shaha, and the principal, as well as Shomaditya for inviting me here to this platform. And I'll be happy to share my thoughts on what we feel is a very exciting era of cancer research. So let me just share my screen. Okay. Okay, how do I get to my presentation here?
You can just select the present now option, Dr. Chakraborty.
Okay. And then a window, is it?
Yeah, a window, and then you have to select the particular PowerPoint presentation or file.
Got it. Okay, there is some technical issue here.
Actually, you have to allow the screen share option. Here, beside the address bar, I guess.
So I can see my PowerPoint, but somewhere it is unable to. It's telling me to go to System Preferences. Then okay. Okay, hopefully you all can see my screen, is it?
Yes, Sayan.
Yes. Okay. So technicalities aside, let's start off with the proceedings. So apologies for the slight delay, and it's really a great evening to be here and to be presenting my research. The topic of my title today, as you may know, is the mechanobiology of Agrin in cancer and tissue healing. And so just a snapshot, what Pritam just inferred about me is that everything has already been discussed. So basically I did my PhD in the US, and I'm fortunate enough to receive a lot of prestigious Singaporean government awards, which I need to thank first before presenting and going on to my research, particularly the ones from NMRC, which is the National Medical Council, and the ones from the Singapore Agency for Science, Technology and Research, which is the government wing for dedicated biomedical research, and the institutional support what we have. And I'm running a special program in these journals, special issue on tumor microenvironment. So any of you budding interested cancer researchers who are willing to send their manuscripts out in these journals, you feel free to just contact me, and I could take it on for there to publish your research in one of these journals as well. And these are my publications, which you can Google out and go in and read about it in more interest if you are really interested to know more about tumor microenvironments. Without further ado, I would take on this opportunity to introduce to you a very basic concept in cell biology. Cells, as we know, or tissues, which are cluster of cells, are indeed surrounded by the extracellular matrix, which is just a protective covering that covers all cells and tissues. Initially, decades of research have identified the ECM as a structural layer that provides support to these cells. But increasingly, it has been considered to be doing much more than just a structural support. Basically, it is regarded now as a more dynamic layer that actively communicates signals to the cells here. So in what ways does it do it? So there are three major aspects by which the ECM communicates its signal to the cells. That is, number one is mechanical stimuli, any form of mechanical changes which it senses by the properties of this ECM. The second is it undergoes active remodeling, and by secreting specific proteins known as matrix metalloproteinase, which cuts other proteins of the ECM. And the third is it basically acts as a sink. So a literally sink that can hold on to various growth factors. And each of these events are transmitted, are sensed to the cells by means of specific receptors which are there on the cell surface. Particularly, I'll be focusing on certain mechanosensing receptors known as integrins, which are very familiar in the cell biology as well as in the cancer, and of molecules of interest to cancer researchers. So what it does is that the cell actively senses these factors in the extracellular matrix. Through these receptors, it is transmitted in the form of signaling events. Each of these signaling events are discretely handled by the headquarters of the cells, which lie in this nuclei. And these nuclei are then actively transcribing genes, altering the behavior of the cell and as an organ itself. So this is a very fundamental aspect of any organ, and more importantly, is actively hijacked by all tumor cells. So much so that the ECM at particular phases of tumorigenesis is actually changing dramatically. And this actually matches the hallmark of a tumor cells as well. This is just a cartoon which we published last year in Nature Materials, which summarizes this event which I just mentioned. So on the left-hand side here, you could see a normal cell is surrounded by, it's more like a normal ECM. But when two of the key factors, one is genetic factors, which are, we all know cancer is nothing but a genetic disease. It's an assembly of a genetic drivers that control the development of a normal to a tumorous state. So these genetic factors, together with these stiffness parameters of the extracellular matrix, which is shown here, so these two are active hits on a normal cells. And when these normal cells are actually bombarded...
Dr. Chakraborty, sorry for the interruption. Actually, slides are not changing. We are just seeing the past slides, but...
Oh, really?
Just share it once again. I think the problem will be solved.
Okay.
Sorry for the interruption.
Stop sharing now?
Stop sharing and share again.
Okay. Is it changing now?
No, just a second. It's presenting. Yeah, it's fine now. It's fine now.
Okay, it's changing.
Yeah, it's working fine. Yeah, it's changing.
Thank you, Shankar. Thanks for the suggestion. So basically, I was talking about the extracellular matrix from this cartoon here. So it assembles three different parameters of the extrinsic environment. One is a mechanical stimuli, which is shown here, and the second is active remodeling of the ECM, which is again here. And then the third acts as a sink by actually holding on to various growth factors. All of these events are actually transmitted inside of the cell by specific receptors, or the mechanosensing receptors, and which translate as signaling responses are sensed by the headquarters of the cells, which are the nuclei. And these then actually alter the gene expression. And so much so, this is fundamental to any cells, but then it is far more hijacked by any tumor cells. And particularly, the ECM adapts to each phase of tumorigenesis and thereby matching up to the cancer hallmarks. So this is a very fundamental aspects of any cancerous cells going on towards forming aggressive tumor. So with that in mind, we look into the various hits that could indeed transform a normal cell into a tumor. So what are those? And to my mind, what is shown in this cartoon, there are two major factors doing it. One is the genetic drivers, which are the genetic mutations that we incur in the normal cell types across our developmental years. And the second is the extrinsic bulk stiffness, or the ECM stiffness as it's called. So basically, when a normal cell experiences these two forms of hits, it is retaliating these inputs by actually changing its material properties. And in the course of time, this becomes a tumor precursor. And then once it is reaching a tumor precursor, its gene expression levels drastically changes so that it further transforms into an aggressive tumor. A tumor is a state of cells where the uncontrolled division occurs and it is beyond any regulation of intrinsic regulations from the cell. So this process of oncogenic reprogramming occurs is you have two important parameters. One is oncogenetic driver mutations, as well as the extrinsic stiffness materials that changes the material properties of a cell. And also the tumor ECM is now drastically corrupted, whereas compared to a normal ECM. So a major motivation of my research here is to identify what are the components of this corrupt ECM, which is presented in many of the solid tumors. So this is the main focus of my research group here in IMCB. So the cancer which I'm focusing on, and I'll be focusing my talk today, is hepatocellular carcinoma, which is a major form of liver cancer, one of the most aggressive known. It is the sixth commonly occurring cancer in the worldwide. We have more than 900,000 cases in 2020 alone. And as you may see, it is of high prevalence within the Asia-Pacific region. Particularly even in Singapore, it's one of the cancers with high incidence rates. So there are several predisposing conditions that make a person susceptible to develop the hepatocellular carcinoma. For instance, a prior exposure to hepatitis B or C infections, alcoholic fatty liver diseases, aflatoxins, as well as chronic injury. So you can think about these are the hits that could actually transform a normal liver, a healthy liver, through a series of events into a cirrhotic stage and finally reaching a cancerous stage. There are some inhibitors or targeted therapies known that could actually help in prolonging the survival of these liver cancer patients. For instance, sorafenib and regorafenib are the standard of care at present. But what you can see from these information is that they only prolong the patient survival by merely 3 months. Of interest, the new kid on the block, cabozantinib, is a multi-tyrosine kinase inhibitor that is known to extend the patient survival by almost 10 months. It's still good enough, but then it's not really best to proceed with these patients in this present line of care. So there is a dire stress in the field to actually find out new targets so that we could develop more novel therapies targeting these various aspects of tumor microenvironment, particularly in the liver cancer. So as I mentioned, the mutational landscape in HCC is ruled by several of the common ones like tumor protein P53, TERT, and beta-catenin being the leading ones that control the architecture, the genetic architecture of the cancer. But the lacuna presently here is the composition of a corrupt ECM. So what is the presenting nature of this ECM that contributes to the development of HCC is rather very poorly known. So here is my research coming in to address at least partially some of the unknowns that are known in terms of HCC ECMs. So the hypothesis of my research group is basically the genetic alterations will definitely give rise to a differential gene expression, and some of the ECM proteins will be absolutely haywire in this process and contributing towards a deregulated signaling events. So basically the objective of this is to identify these altered proteins, and the idea is they will be mechanically conditioning the tumor microenvironment. And what is the benefit of this research? This will lead to the identification of new targets that not only offer innovative lines of therapy, but also offer the opportunities for combinatorial treatments, the ones with existing standard of care. So with this background, a few years back, precisely 6 years back, I did a screen to identify the several overexpressed ECM molecules that are present in these liver cancer lines. Basically, we did a biotinylation of normal liver cells as well as liver cancer cells and identified several cell-associated proteins that are overexpressed in the cancer state, but not in the normal state. And what you can see here in this red cluster is the group of proteins that are differentially attributing to the components of the extracellular matrix that contribute in a hepatocellular carcinoma tumor state. Amongst this list, there were several oncogenes like EGFR, glypican, EPCAM, mucins. Many of them are known to be present in the contributors of HCC. But what was unique in this hit was the protein named Agrin, and it was repeatedly one of our top hits in our screen. So what did we know about Agrin before my research? So whatever it was known is not in the cancer field, I must say. It is identified as a neuromuscular junction protein, a protein that exists between the nerve and the muscle junction. So it is a secretory protein, and it's pretty large proteoglycan of around 210 kDa. Once it is secreted, it binds to its receptors known as lipoprotein-related receptor 4, and binding of agrin to LRP4 actually causes a conformational changes that allows the binding of another receptor named as muscle-specific tyrosine kinases. Together, agrin LRP4 stimulates a phosphorylation event of this protein that leads to the culmination of acetylcholine receptors. And hence it aggregates the acetylcholine receptors, and that's how it got its nomenclature as agrin. So all of this was known in the neuromuscular field. So what is of importance to a cancer field was completely unknown. So the only thing that struck to us was its higher than average expression of agrin in the tumor tissues, particularly in the liver cirrhotic stages, as well as the ones that go on to develop the cancer. So the molecular biochemical architecture of this protein is shown in this cartoon here. What you can see here is it has got multiple heparan sulfate binding domains, as well as several laminin G binding domains. Of particular interest is its C-terminal region. You'll see in my talk that the C-terminal region is highly oncogenic and could actually rescue a variety of oncogenic functions. This is because it has its binding site with its receptors LRP4 as well as beta-1 integrins, which also identified previously by our group that it uses this region to bind to its receptors. So what we comprehend from these analyses was that we first detected that agrin was indeed a protein highly expressing in the liver cancer lines, as well as in the tumor tissues as well. So what you can see from these Western blot analyses, we detected the levels of agrin. These are all liver cancer cells expressing higher than average agrin levels when compared to normal liver cells in the first lane. We did several screens in patients. These are human patients of different stages of liver cancers, and we found that agrin was indeed highly expressed across all stages of cancer. This is again just a nomenclature as showing the various Singapore cohort that expressed high agrin levels in the tumor regions, but not strikingly in the non-tumorous region, suggesting that agrin was specifically secreted by the tumor ECM and is not really present in the normal tissues. So if we analyze several hundreds of patients across the TCGA liver cancer base, and what the red color indicates here is a strong upregulation in the tumor tissue. Strikingly, it is very low in the non-tumorous tissue. All of this points to the fact that since it is very high in the tumor tissues, means it could be readily targeted as well and could pose as an attractive candidate for future therapy. So in this case, we also found out agrin was secreted in various liver cancer lines, as you can see here in the culture media, as well as in the patient's plasma of the cancer patients specifically. Normal patients do not have higher agrin levels as compared to liver cancer patients. Moreover, we also tried to extract these patient tumors and propagate them artificially in the mouse models, which is called as patient-derived xenograft models. And what you can see from these artificially propagated tumor models is specifically the tumor regions denoted by T express high amounts of agrin as compared to non-tumorous surrounding regions as well. So all in all, it is all of this data show that agrin is indeed highly secreted as well as tissue expressed highly within the tumor regions, particularly in the liver. So in the next 10 minutes of my talk, I'll be focusing on how agrin mechanistically promote the oncogenic effects, and that is by altering cancer cell adhesion and migration. Actually, ECM degradation and remodeling is a very interesting trait that is shown by agrin in our studies, and it activates the integrin and the mechanosensing complex, and that regulates the ECM mechanics as well. And I would also shed my light on the YAP mechanotransduction, which is a potent oncogene that agrin activates in the course of its action. And all of these factors corroborate to present a very corrupt HCC microenvironment, which ultimately favors enhanced blood vessel formation within the tumors and leads these tumors to metastasize to lungs. So all of these studies are published in various of my papers, which you can see here, and you can Google it out as well. So in the first case, one of the earlier functions what we noted was its role in cellular motility and invasion. As you can see, when the cells are scratched, they tend to cover up this area by migrating faster. But when we do not have agrin in these cells, this area remains uncovered, suggesting that there is a gross defect in terms of motility. Interestingly, when we are able to add back agrin in these cells, again, the motility is sufficiently restored, suggesting that these are agrin-specific events that is occurring within these cancer cells. There's another depiction by transwell assays where we have shown that agrin presence is actually controlling the invasiveness of these cancer lines. Depletion of agrin, as you can see in the middle lanes, is actually impairing this invasion aspects. And more importantly, when we add back agrin in the culture medium, we could see that there's a subsequent rescue event that is operating in terms of activating invasion of these cancer cells. So the majority of the defect that is attributed in these cells by loss of agrin is actually restored by supplementing exogenous agrin. And this is what you would expect in a dynamic tumor ECM, whereby the ECM is continuously producing a lot of agrin in the cancer microenvironment. More importantly, we also identified several molecular characteristics which were unknown prior to our studies, is the fact that agrin actually controls the levels of invasiveness by migrating to the dorsal ruffling regions as well as the ventral invadopodia structures. On the right-hand side here, you can see these are migrating cancer cells, and one can convincingly say that agrin is actually localized to the leading edge of these migrating cells. So they are somewhat propagating these cancer cells to cover up and migrate faster in order to invade the local matrix regions. And more importantly, the ventral sides of these cells degrade the ECM, and these processes are termed as invadopodia in cell biology. And what you can see here is these black dots, which are regions of degraded gelatin or ECM structures. So what agrin does is helps to promote these ECM degrading structures. In contrast, when you can see when we don't have agrin in these cells, there is a complete loss of these invasive structures. And again, a phenomenon is restored back sufficiently once we add back agrin. So these effects are also shown in our studies in 3D, three-dimensional models as well, whereby we could see these invasive structures emanating out from these three-dimensional tumor-like simulations which we can make in the Petri dishes. So all in all, what these data suggest is that agrin could modulate invasive structures in these liver cancer lines. So how it does, it actually integrates signals coming in from the ECM and transmits them to integrin focal adhesion complex. As I mentioned in my introductory slides, that there are these mechanosensing complexes that senses the extrinsic environment, and agrin actually activates or mechanically stimulates these receptors. And these are shown by these classical adhesion experiments where we don't have agrin. You see a loss of this focal adhesion activity as shown by these Western blot analyses. And more importantly, when agrin is added back here in these settings, you can see that it almost restores the levels of focal adhesions. This event is actually recapitulated by these live cell tracking experiments, whereby in the first panel you could see these are minute, less than submicron structures that is being formed by the development of focal adhesions very early when the cell adheres to the substrate in the presence of agrin here. You could see by 7 minutes there's a strong induction of focal adhesions. What is striking is that when we in these cells which do not have agrin, there's a complete loss or fragmentation of these focal adhesions. And more interestingly, when we have agrin supplemented to these cells, we could again readily form these structures, suggesting that agrin from the ECM is actually serving as a cancer signal to adhere the cells nicely to the substratum and develop and integrate the integrins here as well as with the focal adhesions. So what does all this mean in vivo? All of this data translates to the fact that liver tumors are indeed addicted to agrin expression. So in a classical experiment, we have shown this in mouse models of xenografts as well as in orthotopic in the livers as well. We could see here that in tumors that do not have agrin, they hardly tend to grow, so which is shown by the second panel here. In contrast, when the cells are again rescued back with enough sufficient levels of agrin, you could see that the tumor sizes regain back almost back to the controls. So what it means is that agrin presence or its expression levels bestow enough tumorigenic propensity to these liver tumors. This has been also nicely shown by us in the livers itself, where you could see that the liver tumors that have agrin in them drastically show these nodule-like structures here in these tumors. You can see more than 10 to 12 nodules are present in these livers of mouse. But in contrast, when we are able to deplete endogenous agrin in these mouse livers, you could see only 1 or 2 nodules that is being formed, suggesting that agrin is indeed a key regulator of liver tumor generation and development. So while we were working on these aspects, we were further interested with several of our molecular mechanisms that agrin could control in the liver, ECM particularly. So what we stumbled upon was the Hippo pathway. I'm not sure if many of you know this pathway because this pathway is pretty recent in action, particularly identified almost 10 to 12 years ago. The pathway is highly conserved across mammals from Drosophila to fruit flies. So what this pathway senses is the extrinsic environment, which could be either cell density, organ size, or mechanical stress. And once these are sensed in these cells, a conserved set of serine threonine kinases are activated. For instance, MST, WW41, and LATS1. These are the proteins which are ST kinases or serine threonine kinases. What these proteins activation mean is that they tend to phosphorylate another oncogene named yapentase. Yapentase are paralogs, and they are the converging effectors of this entire Hippo pathway. And recently, yapentase are involved as oncogenes in majority of the human solid cancers known. They are not really implicated in liquid cancers of date, but then all solid cancers, they are the drivers of tumorigenesis. So we hypothesized that agrin might function in this pathway because it is also coming in from outside the extracellular matrix and could find a way to actually nullify these tumor suppressors to actually exert its oncogenic effects on these yapentase oncogenes. So once the oncogenes yapentase are phosphorylated, it is sequestered within the cytoplasm. And this is how the pathway is regulated. If it is in the cytoplasm, it doesn't go inside the nucleus, and hence it doesn't transcribe its set of oncogenic target genes. So the key aspects, what we anticipated was agrin high cells would actually inactivate this pathway. As such, it will promote nuclear yaptase to sustain the oncogenic target gene expression. This is our hypothesis. And we were very happy to find that yaptase were actually also major by that time regulators of ECM mechanics. And this is just in line with whatever story I told you in the past 10 minutes, so much so that yaptase are originally identified as mechanotransduction agents. What it means is that in a stiff cell that is presented by the tumors, YAP is almost definitely in the nucleus as shown here. And this triggers cellular proliferation and oncogenic growth. In contrast, when the cells are confined, localized to small areas, or like a compliant soft ECM, which is anti to that of the tumor, YAP goes outside of the nucleus from this into the cytoplasm. And thereby what happens is apoptosis, growth arrest, and loss of tumorigenesis. And it is increasingly recognized that as the tumor stiffens up, there is a higher risk of HCC with a higher propensity towards poorer outcome. And this is majorly controlled by increased nuclear YAP levels. And of course, featured by several clustered integrin levels as well. So we hypothesized here at this very interesting juncture that is agrin an ECM signal that is relaying the stiff mechanical responses to YAP. And then the question was, how do we do that? So we actually cultured or artificially mimicked these cancer lines to grow them in a three-dimensional spheroids, mimicking soft or hard ECM. What we interestingly found was that when we grew these tumor spheroids in a stiff ECM, we were dramatically increasing agrin levels, as you can see here, compared to the same tumors growing on the soft ones. So what it does mean to us is that, and in this particular phase, we also saw that YAP typically went into the nucleus very nicely, suggesting that higher agrin levels, higher factor activation and integrin and LRP4 receptor aggregation is actually triggering nuclear localization of YAP. So suggesting that there is a strong correlation between a stiffened ECM, high agrin, and YAP function modulation. So through a series of experiments, we were able to show that YAP mechanoregulates YAP in a variety of tunable biomaterials. For instance, in soft matrix versus stiff matrix, also in various 2D hydrogels, which are mechanically mimicking the ECM and its response of the cancer cells, as well as in 3D hydrogels. We were able to show that Agrin regulates YAP in all of these artificially engineered substrates. So this is one such example which I want to just share with you, is that in the soft ECM, YAP is in the cytoplasm, as you can see here, in this particular patterns, and when Agrin is supplemented in these soft ECM or a confined state, we could readily promote nuclear localization of YAP. In contrast, in large patterns where YAP is normally within the nucleus, if we depleted YAP, we could see that Agrin actually able to shift YAP into the cytoplasm, suggesting that Agrin is a true regulator of cell geometry-induced YAP localization. And more importantly, in these depleted cells, if we supplemented recombinant Agrin, we were able to, again, delineate the nuclear localization of YAP. And all of this was actually quantified in these settings here. And not only that, we were able to show this in a variety of tunable soft hydrogels that YAP induces a dephosphorylation of YAP and TAZ, and subsequently a higher target gene expression as well. So it's functionally correlating with the set of oncogenic events that a liver cell is exposed to in these kind of conditions. And there is another image which shows that as we increase the levels of Agrin, we are able to see a gradual induction of nuclear localization of YAP. So suggesting again that higher nuclear YAP within the cancer cells is promising towards a very tumorigenic kind of an environment, and this environment is actually controlled by Agrin from the ECM. So in other studies, we have also correlated these findings in our genetic gene expression studies as well, where we found that loss of Agrin depleted the YAP signature target genes, particularly in the stiff substrates when compared to the soft ones. In contrast, in the bottom panels here, when we supplemented Agrin here in these red panels, we could see that we are sufficiently able to increase the gene expression of the same YAP signature target genes, suggesting that Agrin not only contributes towards nuclear localization of YAP, but also controls the target gene expression and hence the oncogenic phenotype as well. So this is just a cartoon which shows that in the soft setting, which is not very tumorigenic in nature, YAP remains inactive. As the soft setting progresses towards the stiff ones, you could see that higher Agrin levels tend to promote YAP to the nucleus, and hence a more drastic tumorigenic phenotype subsequently follows. And this is exactly what is shown in these data, whereby we have found that the presence of Agrin actually increased the stiffness of the local ECM, the ECM that is surrounding the tumor cells here, what you can see here in these panels. And conversely, when we knock down Agrin in these tumor cells, we could see there's a subsequent compliancy that is achieved in these ECM here. Thereby, they are losing their stiffness here. And that is again reflected very nicely in the localization of actin as well as the YAP-TAZ localization. You can see that the presence of Agrin in the soft ECM is sufficient to promote the nuclear YAP levels. So what this model uniquely proposes is that you can imagine the tumor environment to be a highly stiff one, and that has high levels of Agrin in them. And as such, all of the YAP is present within the nucleus in these conditions. And together, these correspond to a very stiffened kind of tumors, which correspond to very poor prognosis in the patients. So these patients will be living far less longer time points than that of tumors that are less stiff in nature. So this was quite interesting for us to find. And we substantiated our hypothesis in the mouse models as well, whereby we found out a specific YAP inhibitor named vertiporphin. And when Agrin was active in these tumors, you could see that the presence of Agrin was sufficient to enlarge the tumor sizes, as you can see in these middle panels here. And this is just but when these same tumors were treated with a YAP inhibitor named VP or a vertiporphin, you could see that we could achieve several high degree of tumor shrinkage ability, suggesting that targeting YAP activity in these high Agrin cells could serve as an important parameter in actually controlling the tumor sizes and hence the cancer environment as well. And also this data further substantiate that YAP is a major downstream effector of Agrin's function. So this was the first time we were reporting Agrin's involvement in YAP-TAZ oncogenesis, and hence was a very striking discovery on our end. So indeed, all of this data is clinically very relevant because you could see that in liver cancer patients, and these are patients of almost in thousands of patients have been analyzed in this, the patients who have higher than average Agrin levels, as depicted with the red bar here, red line, they significantly live lesser time points as compared to patients who have lower Agrin levels. There's a very clear and significant loss of time. They were like almost months lesser than that of the other group which had higher Agrin levels. And in these spots who have high Agrin levels, they almost sufficiently had almost nuclear levels, nuclear YAP-TAZ, suggesting that in these patients that Agrin levels were sufficient to promote nuclear YAP-TAZ even in the cancer patients. And more importantly, what we found was that in patients that had cumulatively higher YAP as well as higher Agrin levels, they tend to have higher YAP-TAZ target genes as well. And when we grouped these clinical samples according to those target gene expression, we found that there could be a serious consequences in terms of how long they were living. For instance, the patients that had high Agrin and high YAP-TAZ signature targets almost always died earlier than that of those who had less Agrin and less YAP-TAZ target genes. So all of this data clinically corroborates with our biochemical as well as our experimental evidence, what I've shown to you. So in the next few minutes, I would actually focus on the how Agrin choreographs the mechanical landscape of the progression of HCC. So in this way, I have shown that Agrin is a key contributor of cellular proliferation, metric stiffness, topology, as well as invasion. But the one aspect which I did not reveal until now was the vascularization process and the metastasis one. So we also thought if Agrin was important for tumor development, is it really important for tumor spread and angiogenesis as well? And in a way, my next few slides would show an event that Agrin creates a tumor, angiogenic tumor microenvironment that is even more dangerous than the primary tumor environment itself. So for these, we used in vivo assays, which are named as matrigel plug assays, where we could monitor the levels of blood formation within the tumor, early tumor cells as well. And what we found striking was that the presence of Agrin was sufficient to imbibe a lot of tumors within these growing tumor cells. There were a lot of blood vessels that were going inside. In contrast, when in tumors that were depleted of Agrin, we did not find much of the blood vessels intruding in, suggesting that Agrin was a key player for imbibing blood vessels within tumors. So all of this data very nicely corroborates with the metastatic functions that is attributed towards liver cancer aggressiveness. And we could see that in tumors that were like in the first panel, which are controlled tumors, you could see these are metastasizing liver tumors that have gone on to lungs. So this is usually carried through the blood to the distal organs such as lungs. And you could see that in the lung, once they colonize the lungs, there is sufficient degree of blood vessel intrusion within these controlled tumor cells, as depicted in red. Within the T regions actually mark the tumor regions as well. But in contrast, in the mouse that were depleted of Agrin, you could see that there were few tumors that were able to colonize the lungs, but you did not find much of the blood vessel that intruded within the tumors. And as a result, what happened was these tumors were not sustainable and they just died off. So thereby, the tumorigenic ability or the metastatic ability was drastically inhibited in the loss of agrin in this case. In contrast, we had a control experiment where we were able to inject agrin back onto this mouse to again revive the metastatic potential. And indeed, what you can see is these tumors again started to revive back their metastatic ability by imbibing a lot of these red-colored blood vessels from the neighboring regions, suggesting that agrin is actually recruiting a lot of blood vessels from the lungs to help the metastatic tumors to grow. So through a series of experiments, we found an interesting role of agrin in the endothelial cells that actually go and stay within the tumor cells as well. What we found was that the presence of agrin stimulates these endothelial cells to sprout, and that is their major angiogenic property, the way it develops blood vessels within the tumors. And not only that, in the mouse metatarsals in vivo, we were also able to see there were significant degree of sprouting events that was promoted by agrin compared to even more than VEGF, which is a bona fide inducer of angiogenesis. And this was also translated in vivo, where we just planted agrin presence sponges in the mouse skin, and we were very surprised to see that the agrin-containing ones had developed significant levels of blood vessels in comparison to the control ones. All of this data clearly suggests that agrin draws in more blood vessels within the tumors. So how it does so? It actually modulates a very key receptor pathway in the endothelial cells known as the VEGF receptor 2 pathways. So usually there are 3 VEGF receptors known, and it specifically activates VEGFR2. And we found out this by an unbiased phosphotyrosine kinase screen, whereby we found that the agrin depleted cells actually had depleted phosphorylated VEGF receptor 2s. And this is the Western blot showing the same as well. So what we ended up showing in this case was that as the cancer cells secrete high levels of agrin, this is impacting the neighboring endothelial cells in a way that it stabilizes the endothelial VEGFR2, as shown here in this dose-dependent manner when we add agrin in these endothelial cells. Moreover, in the soft matrix, agrin also attributes a significant degree of stability to VEGFR2. And in the stiff matrix, as presented by the cancer cells, the depletion of agrin actually substantially reduces VEGFR2 levels. And this effect could be again rescued by the presence of agrin from the cancer cells. So basically, I'm presenting you a scenario whereby in the soft TCM, we have low VEGFR2 in the endothelial cells, and we have low angiogenic capabilities as well. But in contrast, in the cancer, which has high agrin levels, the ECM stiffens up. This leads to more recruitment of blood vessels, and in the blood vessels, you have higher VEGFR2 stabilization, and this leads to a higher angiogenic or blood flow, and that actually fuels the development of cancer. So what, in a nutshell, I have explained to you is through a series of events, agrin is sensed from the extracellular matrix by the integrin focal adhesion kinase receptors. Their integrity is crucial to transmit the signals to further oncogenes such as YAP and TAZ. YAP and TAZ are activated, they move into the nucleus, sensing this stiff kind of environment, and this translates to an oncogenic transcription signatures. And that corroborates the development of cancer cells into highly invasive and proliferative ones. And not only that, agrin impacts the cancer cells, but in addition, it also impacts the endothelial cells. The improved levels of vascularization, that is denoted by the presence of agrin in these cells, activate the VEGF-VEGFR2 pathways within the endothelial cells, stabilizing them, and thereby this enhances the angiogenic ability or the blood vessel forming ability of the endothelial cells, enabling them to further grow within the cancer cells, and as such it develops to a highly aggressive and invasive tumors, which are also metastatic in nature. So in a nutshell, what I have shared with you is agrin is contributing towards all of these cancer hallmarks by either sustaining proliferative signaling, evading growth factors, resisting cell death, as well as promoting angiogenesis and activation of invasion and metastases. So in all ways, it is a bona fide oncogene that is actually promoting all cancer hallmarks. In the last 2-3 minutes, I would actually shed light on our recent discovery. Most of my talk and my research is focused on tumor genesis as well. But I want to share with you that tumor genesis and wound healing are like brothers and sisters of the same mother. So they basically have a lot of similarities between the events that actually happen molecularly. So as Harold Borak in 1972 first pointed out, that stages of wound healing are actually close parallels of tumor cells as well. Most of the phases of wound healing, such as homeostasis, inflammation, proliferation, remodeling, are also typical characteristics of tumor microenvironment. So like the tumor microenvironment, there are several lacunae that is also present in the wound healing field as well. Particularly, the molecular parameters of the ECM that is lost during wound healing is not known. So obviously, my overarching interest here is Agrin a key player in preserving the mechanoarchitecture of a wound healing niche? And to answer that, we went back to a mouse model and lineage traced these wounded cells in the punch wound biopsies and compared them to the non-wounded mouse model regions. So what we were able to find is an ECM signature. And interestingly, what you can see here is Agrin is one such gene that is increasingly enhanced during the wound healing stages. So the D0 is the day when the mouse is punched and the skin is removed, and versus day 10 is the phase of its healing phase. And what we found along with other ECM changes in the protein is that Agrin is one such protein that enhanced during such expression. And we were initially very excited to see this, and then we followed it up through a timely study that finally functionally delineated that Agrin overexpression in these wound signatures mean. So basically what it does is that it preserves the mechanical signaling, contributes to an actomycin tension in the wound signature, prolongs the development of MMP12, which is a matrix metalloproteinase, and as well as prepares these keratinocytes, or the skin cells, to perceive the mechanical environment much better. And all of this effect is further translated towards improved blood flow within the skin bed, as well as ECM reorganization and MMP production as well. So, as I mentioned, in vitro as well as in vivo mouse models, we did see a strong surge of Agrin expression as we wounded the mouse. And we can see in these brown staining, by 8 days post-healing, when the mouse started to heal, Agrin levels again normalized back, suggesting that its time period of activation was between 2 to 8 days. Through a series of experiments in vivo, we were able to deplete endogenous Agrin in the mouse skin, as you can see here effectively in these Western blot analyses. And what was striking was that in these mouse skin that were depleted of Agrin, these wounds hardly healed as compared to that of the control siRNA, what you can see here within the day 8 of observation. And this was due to a dramatic effect on the impaired keratinocyte reepithelialization, what is shown here in these Agrin-depleted skin sections of the mouse. Through a series of experiments on splinted wound healing models, we were able to see that Agrin indeed affects the keratinocyte reepithelialization, and the loss of Agrin actually halted the migration of these keratinocyte layers that are covering up the wound in general, suggesting that Agrin is a key player in keratinocyte reepithelialization and wound healing response as well. So we were able to replicate these events in the human skin as well. And these skins of the human patients were grown in the lab, and we could see that the depletion of Agrin in these skins left this skin unhealed to a sufficient degree, as shown in the mouse in models in the previous slides as well. And this was again possible due to a loss of keratinocyte reepithelialization in the human skin as well. Most of these effects were again supplemented and rescued with the supplementation of soluble Agrin to these Agrin knockdown cells, suggesting the effects what we are observing are actually specific to Agrin. So a series of biochemical and biophysical experiments, which I would not go into detail, showed to us that Agrin indeed conferred improved traction and stiffness dynamics to these migrating keratinocytes while they were trying to cover up the healing area. So what all of this data truly points out, and this is a patent which I am going to be sharing with you very confidentially, is that which is already we have filed that and is accepted, is we are generating an agrin-based therapy for improved healing effects. And this is true in our mouse models, whereby we could add these recombinant agrin and form of a ointment like an aquaphor or a vaseline, which when added onto the mouse skin, we could see that it has sufficiently high healing capabilities as compared to controls. And this is by improving the mechanoarchitecture of the skin bed, higher than average collagen deposition, as well as promoting the mechanical architecture in the skin wound beds as well. So improved mechanosignaling and blood flow is another trait, what has been shown by our agrin therapy by the presence of improved enhanced MMP12 as well as phosphorylated myosin. All of this level translates to the fact that there is higher than average blood flow within these agrin-treated mouse skin, which corresponds as an outcome of higher healing effects. So what this study actually, which is going to come out in the next month or so in Nature Communications, is that the presence of agrin is a very key and a novel factor of a wound healing extracellular matrix. What it does is that as the skin is wounded, there is high levels of agrin that is secreted out. And this high level of agrin gives resistance to 3 of the bulk rigidity parameters, like the ECM rigidity, force recognition mechanisms, as well as geometry constraints. What these 3 parameters is affecting the keratinocyte migration, allowing their improved mechanoperception, and as such, they are upregulating the cytoskeletal tension, traction stress, and fluidic dynamics. And this is, in fact, accelerating their migratory ability to cover up the wound faster and a higher rate. And all of this event heals up the wound at a faster than average rate. And regarding also the ECM deposition and improved blood vessel formation is also happening in the lower ends of the skin in these mouse models. So what we essentially propose is that the soluble agrin therapy could be an effective wound repair mechanism. And we have uncovered this, quite an exciting story from our end. So with that, I would like to thank our funders, the funding of my grant of the 3 government-funded agencies, Professor Wang Jinhong, who is the ED of our institute, our collaborators in Duke, NUS, as well as in the Mechanobiology Institute, Heike Wohlmann in Germany, Ernest Degucci's team in Mount Sinai in New York, my research group members, and as well as a big thank you for being with me over this journey. Thank you, and I'll be happy to take questions from my end. Thank you.
Thank you, Dr. Chakraborty, for this nice and elaborate presentation. All the best for your future work. And the session is open for discussion and question answers. And the question answer session will be coordinated by my colleague, Dr. Srikanta Guria, Assistant Professor, Department of Zoology, Barasat Government College, and also the Joint Organizing Secretary of this webinar, and our master student, Orpita Shamanth. So over to Srikanta.
Thank you very much for this excellent and very informative lecture. I think there is one question in the chat box from Shutopa Sannal. She asked, is again helpful? I think it is Agrin. Is Agrin helpful for early detection of liver cancer? At what stage of cancer high level of Agrin is noted?
That's a very good question. So we have found agrin across stage 1 to 4 of liver cancer, and levels actually go up from the stage 1 itself. But what is more striking is that we are able to detect agrin at the stage that precedes cancerous stage as well, particularly in the cirrhotic stages. So the levels of agrin tend to be high at the cirrhotic stage, and then it goes up as the cancer starts to develop in stage 1, and then subsequently it tends to go up higher. So that's the rough degree of expression levels of agrin across the stages.
Thank you very much. I request Orpita, please see if any question in the YouTube chat box. I think there is no question in the YouTube chat box. Orpita, please respond.
Srikanta, there is another question from Dhrubajyoti Boiraggo in the Google chat box.
Yes, please, Orpita. present the question of Dhrubajyoti.
Dhrubajyoti asks that can we use the elevated Agrin expression developing any immunotherapeutic regimen against this HCC?
That's a very good question, actually. So, the answer is theoretically yes, but of course we have a lot of constraints in that developing of immunotherapies as well. Because though immunotherapy poses a very attractive measurement, but we have not really elucidated the co-expressing immunogenic cells that is being controlled by Agrin in the context of cancer development. I wouldn't be able to share you the data, but we are working in this field. We are getting there, but I would say that it holds promise, but we are at the stages of nascency. So we still need to wait for a few more years or maybe a few more months to have a concrete answer on that. But the answer is technically yes.
Personally, I have question to Dr. Chakraborty that this Agrin molecule is very much related in the synapse at the times of the release of the neurotransmitter. Because the Agrin is related to the clustering of the acetylcholine receptor. So is there any relation with the Agrin and the neuronal growth or cancer in the neuron? Is there any evidence?
Until now, we have not seen any evidence of neurological cancers activating Agrin in the same way. But what we have seen is that certain pancreatic cancers also express Agrin and could also have higher than average neuronal interference in that way. But until now, we have not seen that direct effect on neurons in cancer by Agrin.
Okay, thank you very much. There is another question from our colleague, Professor Enamul Haq. Please, Orpita, represent the question.
Yes, sir. Sir asked that is there any relationship between Agrin and P53 in gene migration?
Yes, the answer is yes. That's also a very interesting question. And the answer is we have higher Agrin expression contributed by P53 mutations as well. So the loss of P53 might trigger its expression.
There is another question from Mihir Haldar. Role of Agrin is similar in other type of cancer. What is the role of Agrin similar in other type of cancer?
We have looked into liver, lung, and several others. We do see unanimously it's a more similar events in activating YAP, but there are other effectors of oncogenes as well that is being contributed and controlled by Agrin. Of course, I wouldn't share much of the details on that way, which factors, but there are other groups, other oncogenic pathways in other cancers that is also regulated by Agrin, apart from YAPTAZ.
Okay, sir, I have a question.
Yes,
we know that many types of chronic stresses, nutritional habit also are responsible for many types of cancer. And is these types of stresses responsible for Agrin level increasing or that?
We have not really looked into that though, because particularly we are not interested in chronic stress. But what I can say indirectly is chronic stress could be an oncogenic hit by itself. Whether it is regulated, of course, it should regulate a different set of extracellular matrix proteins. It's no surprise there. But then, whether Agrin is one of them, and if it is a direct regulator, we still do not know that. Because it could be an indirect event of some via other protein, which is a stress regulator. But in technicality, it should induce, but we are not sure as to which kind of stress are you referring to and which kind of stress might be the actual trigger of Agrin. That at that point remains yet to be studied. Yeah.
And in case of as Agrin is mainly responsible for liver cancer, as you say, so food habit is somehow related to this, the level of Agrin.
Not that I know of. We have not found any differences in Agrin expression in people who had like like high fat diet or a low fat diet or something like that.
Or alcohol or smoking, is those related?
Smoking is mainly for lung cancer, though. So we have not done those. I do not recall any other groups in the world have done that assay as well until now. As of now, we do not have those data. But then what I can say is that it could be kind of NASH related. So like non-alcoholic fatty liver diseases do have higher agrin. Genetic factors as well as certain kinds of steatosis of the livers would actually upregulate agrin levels. That is prior to cancer development.
And if the level of agrin is increasing in the embryonic stage of a baby birth, is it affect the baby or somehow mother?
Well, so this is a very striking point which you suddenly bring up, and it's a very good question. So the expression of agrin is actually quite dramatic in that stage. So in the before the baby is born, we have high agrin levels, and as soon as the baby is born, agrin levels tend to go down in a normal situation. So it's so down that it's only limited to the neuromuscular junctions. So in the normal tissues, there is hardly much agrin levels. As you would see in the liver also, in an adult healthy liver, we do not have much of agrin as compared to cancer development. So in a way, what it means is that during embryonic development, agrin is actually now shown to regulate heart development as well. So cardiac development is actually controlled in the embryonic stages by agrin. But in adult hearts, you do not have any role of agrin. So because of that, the agrin levels is no longer needed for these adult expression levels. So likewise in the liver as well, for normal hepatocytes, we do not have much of agrin required at normalcy. But it's only on certain diseased conditions like the cancer development, cirrhotic conditions, that these levels tend to rise, and they actually hijack the signaling mechanism somehow. So I would suggest in a way that this is body's intrinsic mechanism to keep or restrain the levels of agrin in normalcy and maintain the normal levels. But in abnormal conditions, abnormal rigidity, stress, or whatever kind of mechanical stress you're looking at, tends to override that effect. And as soon as that happens, agrin levels unboundly goes up. And as a result, the various oncogenic events tend to happen.
Is the level of agrin remains high from the beginning of the pregnancy or after some months or after some weeks?
In pregnant, because the adults, as I say, is low. So only in the fetus it is quite high during the developmental embryonic stages, but then it subsides down normally.
Okay. Thank you, sir.
Appreciate it.
Whether obesity and liver cirrhosis has any direct impact on agrin expression and liver cancer?
Yes, because obesity is known to be a cause of like steatosis in the livers as well as cirrhosis. So basically, in all of these, although we have not really looked into these patients specifically.
Okay, sir. Thank you, sir. There is another, I think, important question from Arpon Roy Chaudhary.
Sorry, I did not get your question. So is this the question? How can we balance wound healing?
Yeah, I am repeating the question, Dr. Chakraborty. That if agrin expression contributes to the wound healing process, then what are the distinctive factors that might play a key role in increasing the tumor?
There is another question from Arpon Roy Chaudhary. If agrin expression contributes to the wound healing process, then what are the distinctive factors that might play a key role? That if agrin expression, then what are the distinctive factors?
So if I understand right, the question is high agrin level and stiffness and formation of blood vessels are important in tumorigenesis, how can we correlate this in wound healing and tumorigenesis? Is there a proper correlation? And the answer is yes. There is a high, because early developments of healing is actually mimicked by that of development of tumorigenesis as well. So in events, what I have shown in the tumorigenesis program could hold of high importance in the healing aspects as well. So basically in that enough period of time, which is the 2 weeks post the wound healing phase, we have a higher propensity for agrin expression. And that could mean that all of these factors are contributing towards an improved healing mechanoarchitecture and the environment. So exactly mimicking that of the tumorigenic environment. But since this is a short-term event,
there is another question from Dr. Enamul Haq. Is anti-agrin therapy is possible in liver cancer?
Yes.
Is anti-agrin therapy is possible in liver cancer?
We have promising evidence as of now, but we are developing that. Yes. The short answer is yes. But we need to be optimistic at this stage.
Yes, then I think there is no question. Urpita, if there is any question in the YouTube chat box?
No, sir. There is no question.
So a big thanks to Dr. Sayan Chakraborty for wonderful explanation and the answering of the queries. I would like to thanks him again, and I request my another colleague, Dr. Somaditya De, to proceed for the next portion.
Thank you, sir. Thank you for this wonderful speech. Thank you.
Thank you. My pleasure.
Thank you, Dr. Chakraborty, once again, and Dr. Guri and Urpita for nicely coordinating the question answer session. So now we are moving on to the next phase of our webinar. And I would again like to request Pritam to introduce our second speaker, Dr. Priyadarshini Chakraborty Basu. She is now actually serving as an assistant professor, Pollinator Health and Apiculture, Department of Biochemistry, Molecular Biology, Entomology, and Plant Pathology, Mississippi State University, United States of America. So, Pritam, over to you.
Yes, sir. Thank you, sir, and thank you, Dr. Chakraborty, for your excellent informative presentation. And thank you to all of you for staying with us. Now we are moving to the second phase of our today's webinar. I am honored to get the opportunity to introduce today's speaker, Dr. Priyadarshini Chakraborty Basu, who will present transdisciplinary approaches to improving pollinator health. Dr. Priyadarshini Chakraborty Basu is currently an assistant professor at the Department of Biochemistry, Molecular Biology, Entomology, and Plant Pathology, Mississippi State University, USA, and is also a courtesy faculty at the Department of Horticulture, Oregon State University, USA. She was a former research associate at the Oregon State University. Dr. Chakraborty Basu received her Ph.D. in Zoology from the University of Calcutta, India. A recipient of numerous national and international awards, Dr. Chakraborty Basu is working on bees nutrition, physiology, toxicology, molecular ecology, functional biology, and neuroethology, using a wide array of multidisciplinary techniques, including multiomics. She is currently serving as the Physiology, Biochemistry, and Toxicology section representative to the Early Career Professional Committee of the Entomological Society of America and chairs the outstanding ECP series subcommittee on entomology today. Now, without any further delay, we return the time over to you, ma'am. Thank you.
Thank you so much, Pritam, for introducing me, and I would really like to thank the organizers for inviting me to this very exciting webinar series. And Dr. Chakraborty, it was an amazing presentation. I really enjoyed listening to your research.
Thank you. Thank you.
I will just quickly try and share my screen. Can you see my screen? Can you see my presentation mode?
Yes, ma'am. Properly.
Thank you so much. So today I am going to talk about a transdisciplinary approach for improving pollinator health. And as I was introduced by Pritam, I am currently an assistant professor of pollination health and apiculture at Mississippi State University. I'm also a courtesy faculty at Oregon State University. I am not sure why am I stuck. So before I start, I just wanted to talk a little bit about why pollinators are important and why this sudden interest in pollinators. Of course, as the term suggests, any organism, be it an insect or a bird, who's involved with providing the very important ecosystem service of pollination is called a pollinator. So as you can imagine that bees, they are very important pollinators because they provide the important ecosystem service of pollination. Thereby, because they are so crucial in pollinating our food crops, they also ensure global food security. And in countries such as the United States, where commercial pollination is a big industry, this can also generate massive revenues of billions of dollars. And not just the food crops, bees also pollinate the wild flora and thereby maintain their biodiversity. And thus, ecosystem stability and food chain stability is also maintained because of bees as pollinators. So this whole idea about, save the bees, save the pollinators, it actually came after, I would say, 2008, 2009, when this became more or less widespread throughout the world. And people became more aware of the importance of pollinators due to this term called colony collapse disorder, which was introduced in the global media. If I show you these two pictures here, to the left, you see that there is this gentleman in Southeast Asia who has climbed up a tree trying to hand pollinate every flower. And if you look at the picture to the right, these are some of our research field sites in California. And these are honeybee hives that have been set out for commercially pollinating the almond trees here. If you just look at the two pictures, and if I ask you that who do you think is a better pollinator, human or bees, the answer is quite obvious. It is a humongous task for a human being to be able to pollinate each and every flower that a colony can in a short span of time. This was another very popular image being circulated in global media. This was from Whole Foods in Rhode Island. So what they did was the picture that you see to your left is a picture in the supermarket with all your produce choices, including food that are pollinated by bees and food that are just grains. And to the right, what you looked at is your produce choices without bees. So just by looking at the pictures, you can see that a lot of fruits and vegetables will be off your diet if there are no bees to pollinate your crops. It is estimated that about 2/3 of what we eat comes due to bees pollinating these food crops. And this is where my research comes in. I employ a transdisciplinary research approach to look into various aspects of bee health. I work on bee nutrition, bee toxicology, bee molecular ecology, bee insect physiology and functional biology, as well as bee neuroethology. So neuroethology is the study of behavior and how the neurons or the brain control it. So in my case, I study how a bee behaves and why it behaves. Even though a lot of my research is basic in concept, it also has an applied perspective because I work with a lot of government agencies, with stakeholders and citizen scientists, and just the general public. So as a researcher, one of my ultimate goals is to be able to translate what I find in the lab or in the field into, easy to understand bits for my stakeholders or for the general public, so that all of us can together work to help improve bee health. And even though I work on pollination biology and apiculture, I also partner with researchers and colleagues working on IPM. They are trying to protect crops. They are trying to work with farmers in providing effective IPM strategies, and that is where I come in. We work together because you will keep on hearing me, say this, that bee protection and crop protection, they are mutually inclusive. So we should be able to protect our bees as much as we should be able to protect our crops from pests. And this is also one of the research interfaces that I collaborate on. Just to give you a brief introduction to my background, my PhD was from the University of Calcutta. I was a DST Inspired Fellow and a gold medalist from the department. And I worked across 4 Indian states looking at 2 Indian species of honeybees, Apis cerana and Apis dorsata. So I studied the impacts of pesticides on these bees. I was also a Newton Bhabha Fellow, and I worked at the Institute of Neuroscience in Newcastle University, where I studied the gustatory perceptions in bumblebees exposed to pesticides. From there, then I moved on to Oregon State University as a postdoc, and I worked on my 4th type of bee species, Apis mellifera. And from here, I moved to Mississippi State University, where currently I'm a tenure-track faculty working on bee health. And I also have research collaborations across North America, Europe, as well as still in India. When you think about what is causing bee decline, it is not an easy answer. There are multiple factors that are affecting the global bee populations. And all of these stressors, all of these factors either work alone or in synergy with each other to cause further stress. So you can have pests, parasites such as varroa mites, nosema, beetles, wax moths. You also have pathogens, a variety of viruses, bacteria, and fungi. You have poor nutrition due to monocropping and habitat loss. Of course, pesticides, which is probably one of the most talked about stressors. Environmental stress, stress due to repeated transportation to meet the pollination needs across different commercial crops, and also genetic bottlenecking. So as a beekeeper, you may be using the same genetic stock over and over and over for years, for generations. That can also cause a decline in the quality of bees that you are producing. And this is what I look into. I look into understanding how all of these stressors, either alone or in synergy with each other, affect bee health. I lead the Mississippi State University Pollinator Health and Apiculture Lab, and my lab has this overarching theme for research whereby I look into the impacts of poor nutrition on bees. I also study the impacts of pesticides, of xenobiotics on bee health. I look into how the environmental stressors can impact bees. And of course, as I mentioned, how all of these stressors, these multiple stressors, either act alone or in synergy with each other to cause further stress. Due to time constraint, I will not go into details of each of these sections, but I will pick and choose a couple of studies or maybe a few studies to talk about for poor nutrition, pesticides, environmental stress, and multi-stress in the course of my presentation. And I understand that there are students present in today's webinar, so each of my slides is numbered to the right bottom. If you think I'm speaking too fast, or if you think that, there are some concepts that I haven't explained enough, I have tried to keep it as simple as I can. Please reach out to me with questions at the end of my presentation. Let me first talk about nutrition. I am one of the leading researchers on bee nutrition, currently not only in the United States, but also globally. So I look into how the pollen nutritional quality of commercial crops and any of the non-crop pollen forage affects bee health, bee physiology, colony health. I look at what are the different nutrients, the macronutrients and the micronutrients that are required in bees based on their different life cycle stages. What is the availability of the various diverse forage for bees in a landscape? And of course, how do all of these nutrients shape bee gut microbiome? So I try to improve pollinator forage and pollinator habitats by examining not only the forage choices that bees make, but also the nutritional quality of the habitat. And I am now currently trying to build the first ever database for nutritional landscape that can be made available to all bees across North America. And I will talk a little bit about this in some of my subsequent slides. I also examine how these nutrients shape bee physiology and bee functional biology. It could be honeybees, it could be native bees, but how do these nutrients shape the physiology in these bees? This is also something that I look into. So there are many ways that bee colonies can face nutritional challenges. There can be a lack of pollen abundance. The quantity of pollen that is coming into the colony is just not enough. There can also be a lack of pollen diversity. Too much of only one type of pollen is coming in. Think about it from the human perspective. Suppose if you are only asked to eat rice every day, you are not getting optimal nutrition. If you're only asked to eat meat every day and no veggies, that's also not enough of nutrition that you're getting. It's the same with bees. They need enough of pollen, but they also need a diversity of pollen. So another way that a colony can be nutritionally challenged is that the pollen that they're bringing back in is of inferior quality. For example, we have seen that blueberries and hybrid carrot seeds, these plant pollens are not as nutritionally optimal as some of the other pollens, such as almonds or such as apples. There can also be an insufficient nectar flow in the landscape, not enough plants producing nectar so that the bees can bring it back into the colony and make honey. And of course, winter survival. If you have a beehive, your bees need to be well fed, they need to have better nutrition, better nutritional immunity to be able to survive throughout those winter months and then help rear the next generation of brood in the following spring. So overwinter survival due to poor nutrition is also a major concern. So one of the research areas that I lead is understanding the sterile nutritional physiology in honeybees, and not just honeybees, but also in native bees. So phytosterols, think of it as something comparable to the cholesterol in human beings. These are essential micronutrients. They are a certain form of lipids, and they are present in plant pollen, and bees really need them. And I will show you in a later slide why phytosterols are important for bees. So as I was mentioning that commercial pollination in the US, it generates a tremendous amount of revenue. For example, if we are just looking at the almond pollination in California, it can generate anywhere between $11 to $13 billion in revenue. However, to be able to pollinate all of these almonds, bees are forced to start their brood cycle earlier than they usually would. And the US beekeepers, they are certainly concerned about bee health, and we all understand that there is a need for better nutrition. The picture that you see to the right of the screen is what a typical commercial supplement looks like. So during times in the landscape when there is not enough flowering plants and bees are not getting pollen, this is what beekeepers will feed to their colony. This is a commercial protein supplement that is given to the honeybees. So studies have shown that colonies which are only given these commercial supplements and no pollen, they do not sustain well as good as colonies which have plenty of pollen to source from in the landscape. And in fact, a major focus for decades has been on macronutrients such as proteins and carbohydrates and how they shape bee health. Research on micronutrients such as phytosterols is severely lacking. When I first started working in this area and I was looking into, the existing research that we have, I realized that probably the last few papers that were published were, I believe, from late 1970s and early 1980s. So there certainly has been this huge gap for decades in research on understanding how micronutrients can shape bee health. So let me show you this quick animation video as to why sterols are really important for bees. Pollen naturally has an abundance of phytosterols. When bees consume pollen, they can take up these phytosterols, and these phytosterols are stored in the body tissues of nurse bees as endogenous sterols. These endogenous sterols are then transferred to the growing larva through brood food. Sterols are required for producing molting hormones. So larvae have sterols, hormones, and then now you have successive generations of bees. If these commercial protein supplements lack these critical phytosterols, after a point of time, even if bees are consuming this, they have no new sterols to replace their endogenous body tissue sterols. No sterols going into the larva, no molting hormones, and after a point of time, no new generation of bees. And that is why phytosterols are really important. So not just phytosterols, but also with other nutrients, what I do is I make these artificial diets, or I play around with different types of pollen. I dose bees to these different diets in the lab, as well as I dose smaller colonies in semi-field and large-scale field studies. And I conduct various multi-omics analyses to look into how these different types of nutrients are shaping bee physiology. So I conduct targeted lipidomics to look into phytosterols. Lipidomics, this type of analysis gives you exact quantification of the different types of lipids that you're looking into. In my case, I look into phytosterols. I also conduct untargeted metabolomics, whereby I look at, imagine this to be like a snapshot of all the metabolites, the biomolecules, the small molecules, the different amino acids, for example, that are present in the bee tissues. And I also conduct proteomics, which gives us a snapshot of all the proteins that are present in the bee tissues. Following up on this multi-omics approach, I also conduct all of these multi-omics analysis on pollen, plant pollen, to look at what is the nutritional resource that the pollen grains provide to the bees. And I call this project the omics approach to bee nutritional landscape. Currently, habitat for bees are chosen based on their relative attractiveness, but not on their nutritional quality. So we choose a plant for bee habitat because we see that a bee could be a bumblebee, could be a honeybee, could be a stingless bee. These bees, they are attracted to that particular plant. So that is why we are choosing to plant it. But do we really understand what is the nutritional quality of its pollen? What is the nutritional quality of its nectar? And what is it actually providing to the bees? No such information on the micronutrient profiles are available for plants in North America, which is why it is important that we understand the nutritional profile of not only just the commercially pollinated crops, but also all of the natural forage that is available in a bee's habitat. And I also want to look into the nutritional quality of these commercially available diets, a picture that I showed you a couple of slides ago. So what are these commercial diets providing to the bees? That is also something that I'm looking into. And I have been very fortunate to be funded by the US Department of Agriculture to actually conduct this study. So right now I am trying to build a database for understanding the pollen nutritional quality of over 100 major crop and non-crop plants in North America. And through this, we can provide feasible practical recommendations for growers, for beekeepers, for researchers, for policymakers, as well as just for concerned citizens. If you might be a homeowner and you have a backyard or a little garden, through this database, you can choose which plants that you can plant to promote bee health by providing better nutrition. So currently, I'm looking into the protein contents, the lipid contents, the amino acids, sterols, metabolites, and phytochemicals that are present in the various types of pollen in a bee's habitat. As I mentioned, I conduct targeted lipidomics, and I'm collaborating with the Oregon State Mass Spectrometry Center with my colleagues there. And I believe we are the only lab in the US who are actually working on this, and we can exactly quantify about 12 to 13 different major and minor phytosterols across a wide variety of sample types. We also conduct untargeted metabolomics, and we look into the amino acid profiles. And as I mentioned before, we are now looking into the diversity of forage that is available to bees and looking into understanding what is the nutritional quality of this forage that bees are getting access to. The second, another major area of research focus in my lab is understanding the impacts of xenobiotics on bees. So I try to estimate the physiological responses in bees to field-realistic multi-pesticide exposures. And I emphasize on this because in the lab, you can just randomly select a pesticide and a dose, and you can dose your bees. However, in the field, the scenario is quite different. You have a different dose exposure. You have a multitude of pesticides that bees are exposed to in different ways that bees are exposed to. So this is something that I look into. I also try and understand the impacts of pesticide synergies. How do different groups of pesticides work together? Is one combination more toxic than the other? As I mentioned before, I work with my IPM partners to understand the efficacy of their IPM techniques and if that is safe for bee pollinators in the field. And we try and work together formulating alternate IPM strategies. So for example, what time are they spraying? What are they spraying? Are they spraying something on the tree itself? Are they spraying something on plants in the understory? What type of bees are being exposed to these? And what doses of exposure are they receiving in the landscape? And I conduct these studies just not in the field, but also in the lab. So I examine these individual impacts to assess the various colony health measures, as well as individual bee health measures across the various castes and life cycle stages in honeybees, also across the various life cycle stages in native bees. So the first example that I wanted to show you, talking about impacts of pesticides on bees, is this study during my PhD, where I looked into the impacts of field-realistic pesticide exposures on Asian wild honeybees. In 2011, when I started doing my PhD, a baseline data was missing from the Indian subcontinent. There was no other bee lab, there was hardly any bee research looking into the impacts of field-realistic multiple pesticide exposures. In fact, not just the researchers, the government ag offices, they also needed to be able to establish the status of pesticide use as well as their impacts on wild honeybees. So I looked into the field-realistic multiple pesticide exposures and their impacts on Apis dorsata and Apis cerana. Apis dorsata is, you have seen these bees. These are the giant honeybees. They build these massive combs hanging from the edge of a building or from a tree branch or even from like a rock crevice. Apis cerana, on the other hand, are smaller in size. Some beekeepers tend to domesticate them by keeping them in bee boxes, or you can also find them naturally inside tree hollows, but these are cavity nesting bees. So through my PhD research, I was able to identify exposure doses which were safe in the field, beyond which I was observing both lethal and sublethal impacts. Lethal impacts indicate that if your bees are exposed to this particular dose of pesticide, they die instantly. Sublethal impacts, however, are more dangerous because even though the bees are not dying immediately and you don't understand that there is a problem, these bees are already exposed to pesticides. They are taking back the contaminated nectar and pollen back to the hive. The rest of the colony is feeding on it, and sublethal impacts can affect their physiology, their behavior, their cognition, their overall colony health. And before you know, it's already too late and your bees have died or their population has declined. So it is really important to be also able to identify not just the lethal effects, but also the sublethal effects. And this research project that I was involved with as a larger project in my PhD labs, we had collaborations across India and UK with various researchers, non-profit organizations, stakeholders, as well as government ag agencies. So we found that both in the field as well as in the lab, exposure to pesticides increased the oxidative stress in bees. And as the dose increased, as we expected, the oxidative stress also increased. So important antioxidant enzymes such as superoxide dismutase, catalase, oxidative stress markers such as xanthine oxidase, all of these were significantly higher in bees that were exposed to higher doses of pesticides. We also found changes in bee olfaction and their visual acuity. So how were bees responding to their sense of smell, to their sense of vision? All of these were significantly affected due to pesticide exposures, both in the field and in the lab. We also conducted scanning electron microscopy on bee antennal sensilla and their compound eye ultrastructures, and we found morphological deformities in honeybees which have been exposed to pesticides over a long period of time. And their neurophysiology was also altered. So I would live dissect bee brains, and we would expose bees to pesticides. We would give them also an odor and stain that live bee brain with a combination of two pesticides. With a combination of two stains. Fluorotodextran, if you see here on this right panel, the green stain, it fluoresces when it combines with biologically active free calcium. And I will talk about this. Tetramethylrhodamine dextran is this red stain which basically binds with the whole tissue mass and then it fluoresces red. And the third panel here is a merged panel which is basically showing you what these two stains look like together. So what we did was I dissected these bee brains and we stained them with these two stains. Biologically active free calcium is very important for insect olfaction. So more the calcium, better the olfaction, more the staining in the bee brain. And what you see here in this right image, that the control bees have higher green stains compared to the pesticide bees, which essentially tells you that why their olfaction was hampered, because they have less staining and less free calcium. And we then did these optical sectioning of 5-micron diameters of the brains. And this is what all of the sections put together into a Z-stack looks like in this little animated video. And we also looked into the various calcium handling proteins such as calpain-1, as well as important visual proteins such as rhodopsin and opsin. And we found that all of these proteins were also significantly downregulated when bees were exposed to pesticides. So this definitely tells us that not just bee physiology, bee behavior, but also their neurophysiology and their ultrastructures were impacted, were changed, were altered because of pesticide exposures. Another study that I'm currently involved with is understanding the impacts of pesticides across the different age cohorts of adult honeybees. So EPA, the Environmental Protection Agency here in the United States, they have very strict and stringent regulations for assessing toxicity of various pesticides to bee pollinators. However, these are done across 3 stages or 3 tiers. Tier 1, which is an exclusively small-scale lab study. Tier 2, Tier 2, which is like a semi-field study. And Tier 3, which is a large-scale study. And this is hardly done. Mostly, all of these studies are done on 2-day-old honeybees. So you can imagine that in a colony, for example, in a honeybee colony, you have eggs, you have larvae, you have different ages of the adult. So conducting these studies on only 2-day-old adult honeybees was something that we thought, well, what happens if you have a different age cohort of the adult bee? So what's happening with them? To understand that, we have conducted this large-scale study where we have, collected different age group of bees and we expose them to pesticides. And we are finding differences in how these different age cohorts are responding to the same pesticide exposure. So we worked on 24-day-old foragers, we worked on 10-day-old nurse bees, we also looked into these impacts on newly emerged adult honeybees, which were less than a day old. And the paint markings, so we paint thousands of bees for different experiments. The paint markings helped us identify what age group the bees were in. The third major research area is where I try to understand the physiological adaptations in bees due to the various environmental stressors. I will only show you one study in this area, the impact of the wildfires on honeybees. If you are not familiar with US geography, so we have, like in India, we have the West Coast, we have the East Coast, we have the Midwest, the Southeast and the Southwest, and that sort of, like the Pacific Northwest, the New England area. So there are different areas. The West Coast in general is suffering through extensive heat waves over the last many years, extensive drought in summer, and as a result, severe wildfires. I lived in Oregon for about 5 years, and not a year has gone by where we haven't been affected by wildfires, and it is only getting worse each year. So this was a massive wildfire event that occurred last year, and unfortunately, a lot of people lost their lives as well as their property, and we lost millions of acres of natural vegetation due to these wildfires. This was a devastating natural phenomenon. In fact, this wildfire was so intense that the smoke from these unprecedented West Coast fires, they not only moved across North America, across the Atlantic, they moved all the way from the Pacific coast to mainland Europe. That smoke was so intense. So what you see here to the left, these two images are shared to us by one of our very close collaborator, an Oregonian beekeeper, and he's also nationally recognized. So Mr. Hansen, he had hundreds of beehives that were stored in these various holding yards, which were close to, unfortunately, areas that were affected by wildfires. So this image to the left, where my cursor is, you see him standing around burned patches where his hives were. All of these hives were burnt down by the wildfire. To the right, you also see these burned patches. Miraculously, some of his hives survived the direct contact with wildfire, and some of his hives did not. So you can see the burned patches, while some hives survived. However, these hives, as you can imagine, were exposed to extreme heat from the wildfires. Even though they were not directly in contact with the fire, they were exposed to this extreme heat. This video to the right that you see playing is shared to us by another close collaborator and Oregonian beekeeper, Tom Simpini. So Mr. Simpini's hives, they were also in this holding yard away from the main wildfire site, actually hundreds of miles away from the wildfire site. And this is very close to where the Oregon State University campus is, I would say about 20 miles from the campus. And what you see here is this intense smoke and red haze from the wildfire. Mind you, there is no fire burning right next to these hives. The fire is burning at least 50 to 100 miles away, the nearest fire. And what you see here is the smoke and the intense red haze. So even though these bees, they were not exposed to the heat or to the direct wildfire, they could not go out. They could not pollinate. They could not fly because of this intense smoke and this red haze that you can see. It was so dangerous because of the smoke that even us as humans, we were asked to stay at home for 7 days at a stretch. We were asked not to go out. We were asked to use filters at home, use respirators for breathing because of the particulate matter and the ash in the air. And these two images are from the town that I lived in Oregon. And you can see I'm away from the wildfires. And yet, look at that intense smoke and that haze. And this was probably like maybe 11 o'clock in the morning. And I can assure you, in person, this was devastating. I mean, this was way more dangerous than what the pictures look like. And these were some of our research hives that we had set out close to campus. And as you can see, this intense smoke. So what we did was, as researchers, we environmental conditions. So we decided that we have these research apiaries that are set out. We have a research hives. Let's conduct a study and look at what are the impacts of wildfire and smoke on these honeybees during this wildfire event. So we sampled bees from some of the research colonies during this intense wildfire smoke, and we sampled bees from these colonies a month after the smoke when everything had cleared out. And this is my postdoc mentor from Oregon State and my present collaborator, Dr. Ramesh Sagili. As you can see, he had to wear a respirator to be able to go out and collect these bee samples, and even then he was struggling to breathe. We also collected honeybee queens from colonies which were directly affected by the wildfire itself. And we are dissecting out the spermatheca. You can see me here dissecting a queen spermatheca. And we are looking into the various morphometrics of the queens. We are also conducting proteomic analysis on the queen spermatheca, trying to look at how the reproductive quality in the queen might have been affected due to direct exposure to the wildfire heat. We have also sent our spermatheca samples to our collaborators in NC State University. And they are looking at the various queen morphometric measurements. They're looking into the spermatheca. They are looking into live sperm and the dead sperm in the spermatheca. And they're trying to analyze and see that if these queens, they were exposed to either direct wildfire heat or not to the heat from the wildfires, what's the status of the sperm in their spermatheca and what's the status of their reproductive quality. So this study is still currently under progress, and as you can imagine, we have millions of samples to look into. And finally, for the last research focus area, I just wanted to quickly show you another study whereby we are looking into the interactions between the different stressors. As I mentioned before, that my lab also looks into how these different stressors, these multiple stressors, they interact with each other. Are they working alone? Are they more harmful alone, or as a crew in synergy, they are more detrimental to bee health? And we also try and formulate various mitigative strategies to be able to counteract these stressors. So I look into how they shape and impact pollinator physiology, their behavior, colony health. So this particular study was done with our same collaborators in North Carolina State University, where we looked into the impacts of multiple pesticide exposures on royal jelly quantity as well as the nutritional quality of royal jelly. Those of you who are not familiar, just like a couple of quick sentences on bee biology, like a crash course. So in honeybee colony, the nurse bees, the young adults, they feed the entire colony. They have these glands, the mandibular gland and the hypopharyngeal gland in their head region, whereby they produce these proteinaceous secretions to feed to the rest of the colony. So if they are making brood food, which is nutritionally of lesser quality, they are feeding it to all the larvae who will become workers. If they are producing a very high quality diet called royal jelly, this is only provided to the queen larvae. So what we wanted to do is we exposed certain colonies to multiple pesticides through these pollen patties that you can see in this image here. And we reared these queen cells. We then harvested the royal jelly. What you see here are these tiny little queen larvae in these queen cells floating in a pool of royal jelly. So we harvested these royal jelly samples, and we wanted to see, firstly, if there is a difference in the quantity of royal jelly that is produced because these colonies, they were exposed to pesticides compared to the control colonies. And secondly, we wanted to understand if the nutritional quality of the royal jelly samples varied because of exposure to pesticides. And we found a difference in both. So we found that the quantity of royal jelly that was being produced in colonies that were exposed to pesticides was lower than the quantity of royal jelly that was being produced in colonies, control colonies, which were not exposed to pesticides. We also found that the phytosterols, the metabolites, the proteins in these royal jellies significantly altered when colonies were exposed to pesticides. So this certainly tells us that pesticides, beyond just immediate physiological impacts or immediate mortality in honeybees, they can also cause nutritional stress. And I just wanted to show you a couple of quick slides as to what else I do other than research. I am heavily involved with teaching and mentoring students, and as you can make out, I work across the world. So my work is really not defined by the boundaries. I regularly mentor students both at Oregon State as well as Mississippi State. I'm also developing courses on insect behavior and new ecology, as well as honeybee biology, pollination biology here at Mississippi State University. And a lot of these courses will also be offered online. So if you or any of your students are interested, you can reach out to me to know more about these online classes that we offer. I'm also heavily involved with outreach and extension because, as I mentioned, that I am very cognizant of how research can be translated to serve not only the stakeholder needs, but also with our extension partners, our collaborators, general public, concerned citizens. So my teaching, mentoring, outreach, and extension efforts are, as I would say, not limited by any boundaries. And as you can see, I've worked across in India, in UK, as well as across North America. With that, I would like to thank all of my mentors, my collaborators, my research extension partners, both the OSU Honeybee Lab and my Pollinator Health Lab at Mississippi State. We are expanding. We are getting new students soon. And also my husband, Basu, for all the help and the support that they have been providing me throughout my career so far, and they're continuing to provide me with help and support. Without any of their help, none of this would have been possible. And I would also like to thank the organizers once more for inviting me to this very exciting webinar. Thank you so much for patiently sitting through my presentation. If you have any questions, I'll be happy to answer them now. You can also reach out to me at pd1090@mstate.edu or basachat@oregonstate.edu. And I hope you stay safe amidst all of these uncertainties during the pandemic. Take care and please stay safe. Thank you.
Thank you so much, Dr. Chakraborty Basu. Thank you for this nice and amazing presentation. Now I would like to request Dr. Srikanta Guria as well as our MSc Century student Soumya Mishra for the question and answer session. Now the session is open for discussion. Soumya, you may start. There are
Thank you very much.
Questions.
Thank you very much, Dr. Priyadarshini Chakraborty Basu. There are some questions in the chat box. I would like to present the first question from Dr. Jayati Ghosh. What are your suggestions for beekeepers, especially in countries like India, where chemical pesticides are in common use?
That is a great question. And I have to say that we have been struggling with that ever since I did my PhD. I think from my perspective, there could be various approaches to working on this problem. As I keep mentioning that bee protection and crop protection, they're mutually inclusive. So we should be able to protect our bees as much as we should be able to protect our crops from insect pests. So I think the first, I would say, approach would be to create a proper network of beekeepers and growers, have an open channel of communication, spray pesticides when bees are not foraging or when the flowers are not in bloom. Study the pesticide labels very carefully and work with the grower. The beekeeper and the grower, they need to partner together in this problem. So if you are reading the pesticide label carefully and you look at the residual toxicity, for example, for product A, it is no longer toxic after 8 hours. So maybe try and spray it at 6 o'clock in the evening. So by the time it is 6 o'clock in the morning and the bees are out foraging, it is no longer toxic for bees. Avoid sprays during peak bloom when bees are out there. Avoid direct sprays in and around the colony. So there are some of these ways that you can potentially protect bees from pesticide exposures in the field.
I would request Soumya Mishra, please present the question of Professor Sumana Shah.
Thank you so much, ma'am, for this very informative session. So the question which is asked from our HOD, ma'am, is there any data that cell phone radiation affects bees and their pollen carrying behavior? Yeah.
That is a great question. And believe me, Dr. Saha, I get a lot of the same questions from our beekeepers and environmental groups here. To be very honest, there is no study and there is no data available right now. Pollen carrying capacity. So I do know of one recent study from Oregon State University where they exposed zebrafish larvae to 5G cell phone radiation and they found no adverse effects. So this is the only study that I know of regarding radiation and an organism, even if not bees. But other than that, I have not come across any published study or research that has looked into this aspect.
Okay, there is one more question which is asked from.
No, there is another question from our HOD madam
Exactly.
What are common pesticides disrupt brain functioning in the bees in your place?
So there are a couple of pesticides that I have personally worked with, and we have found that. So those were organochlorines, pyrethroids, synthetic pyrethroids to be specific, and one endosulfan.
Yes, another question from our student Pooja Upadhyay. What are the direct effects of the forest fire on the pollination process by the honeybees?
That is a great question. So the direct effect of the fires, I would say, could be many. So for example, we need to look at it from two perspectives. One, the fire reached the honeybee colony or the fire reached the native bee nest and it burnt everything in its path. So of course you'll have dead bees, dead honeybees. The other could be the fire did not reach it, but there is intense heat. The colony can choose to abscond. They can, leave their colony. They can fly to a nearby tree branch and they can just hang and sit there. Some people have spoken, some beekeepers have shown that this is something that they have seen. The main problem with this particular behavior is that because it's a sudden phenomenon, bees are not prepared. If it was a swarming event, bees would have been prepared, with their diet, with their extra food reserves and everything. But because it's a sudden environmental phenomenon, the bees are not prepared. If they are abandoning their colony and they're choosing to swarm or aggregate elsewhere, they are going to starve because everything else around them have burnt and there is no other food resource. The third scenario could be that the bees are not in the path of wildfire. The bees are also not in the path of direct heat. But as I showed you, they are far away from the wildfire event and they are covered with smoke and this red haze. In that case, they cannot fly out or pollinate because bees are dependent on the plain polarized light from our sun to be able to match their visual landscapes and fly. So if they cannot do that, they can't fly out. Even the smoke, in a couple of studies with butterflies and caterpillars, they have shown that smoke can affect bee health as well, the insect health as well. So there are, I would say, many ways that the forest fires can affect the pollination process. I wouldn't say as a pollination process, but generally a forest fire would occur in a forest far away from where the crops are. And the particular season that these forest fires occur here in the West Coast, we have fewer crops that need pollination. So what it can actually affect is how bees are able to fly and what their colony status is like.
Yes, Somo, please ask question from Pritam Chakraborty, Ritam Mondol and Orpita Samanto.
Okay. So, Pritam said, is there any direct effect of repeated smoke exposure on fire alarm, pheromone of bees like isopentene acetate?
That is a great question. It may have. Nobody has really looked into it, and that is why I keep on emphasizing that we hardly know what the effects of these wildfire and smoke events can be on bees. But if I just talk about general beekeeping practice, we use a smoker to mask these alarm pheromones. So when you're opening up a honeybee colony and you're working, with that colony, these bees will come and try and sting you, right? So we use a smoker, we use pellets and other fuel. We create artificially the smoke to be able to mask these pheromones, just so that the bees are kind of a little disoriented. They try to stay away from us, or they try and go back into their colony, and they don't sting us. So this just could be that repeated smoke exposures will, of course, mask their ability to communicate with each other.
Okay, the next question is from Preetam Mandal. He is asking what type of home and cost-friendly food supplements will be healthy enough for bees for rural, economically weaker beekeepers. Yeah.
You can do two things. You can plant a diversity of forage around your colony. So suppose where you're placing these colonies as a beekeeper, you can have it in a holding yard. Depending on your local planting regulations and guides, you can plant some mustards, or you can plant some other types of forage that can be beneficial for the bees. Second thing that you can do is, we have something called a pollen trap that you can place on a colony to trap for pollen, and we use it for research. As a beekeeper, if your colony is at that particular time of the year when it's bringing back a lot of pollen, you can choose to trap for these pollens, store them in your house, and then you can just make a paddy out of it and give it to the colony itself during times of pollen dearth. So all you then invest is just installing these pollen traps, and that's it. You're using exactly what bees would choose to take in a different time of the year, and you're giving it to the bees when they don't have access to enough pollen in the landscape.
Okay, thank you, ma'am. The next question is from Arpita Shumanta. She's asking, as we all know that honey has an anti-aging effect, does honey help genetically or superficially to improve aging-related problems?
That is a great question. Unfortunately, that is not something that I work on, but I have heard of people anecdotally saying, or also some of the studies that have come out looking into the properties of the various components of honey and their anti-aging effects. But unfortunately, at this point, I don't know if honey can help genetically or superficially to improve aging-related problems. I mean, there might be cosmetic companies cashing in on this, but this is not something that I work on. I work with honeybees. I work with the organism.
Thank you very much. Another question in the YouTube chat box from Rebonto Roy. Did you get any evidence which suggests any change in the dynamic time of pollination and even in the nectar consumption of bees after the widespread wildfire in the US?
Very interesting question. So just to give you a context, thank you for that question. I should have explained it a little better. The wildfire events usually happen peak or late summer. At this time, it is like, I would say, end of July or, end of August or September. So this event that I was showing you, it happened around middle of September last year. At this time, there are hardly any crops which need commercial pollination. So the bees were all being prepared for overwintering for fall, and they were already being moved to the various holding yards. So because they were already at a particular holding yard and they were not pollinating, there is really no before and after to compare. And in some places where these bees, they were directly in that wildfire zone itself, we could not even access the hives for up to a month because the Forest Service had closed off the area. The fire department, they were working tirelessly day and night to save human property and the forests from burning further. So as a result, in some cases, we did not even know what has happened to the colonies before a whole month because we just could not access that area. Everything was burned down and quarantined.
Another question asked by Professor Enamul Haq, are bees developing resistance against pesticides?
Yes. I wouldn't call it resistance as such, but this is also something, it's like a work in progress. So I really don't have like a yes or a no answer at this moment. but it would all depend on how long you're being exposed to pesticides and then what their life cycle is like. So if you have a colony in a landscape that has been there for generations, they may develop certain mechanisms to be able to adapt to the pesticide exposures. One of my past research has actually looked into exactly this. So we followed honeybee colonies in a pesticide-intensive landscape for months, for about that particular colony, which were constantly being exposed to pesticides, and we found that some of their some of their pesticide detoxification enzymes, like esterases, they were becoming more diverse. So they were showing a certain degree of heterogeneity to be able to counteract such pesticide exposures compared to the colonies which were not exposed to pesticides for the same duration of time in a natural landscape. So I wouldn't call it pesticide resistance. I would rather call it adaptive measures that a colony can take if they are being constantly exposed to pesticides. But they have to survive through all the exposure events. They have to be in that landscape for months at a time, across generations, because this is not something that can happen overnight. But great question.
Soma asks the question from Doipayon Haldar.
Okay. The question is it possible to use drone or other instruments for pollination instead of bees?
That's a very interesting question. Actually, Walmart and a few other companies, they are now trying to come up with the drone technology for pollination, for commercial pollination, trying to see if robots can replace bees. It's not that easy. There are multiple semiochemicals and cues that occur between flowers and bees, which ensure pollination. And I personally, I don't think that drones will be able to pollinate crops as effectively as bees can.
Another question that is very much interesting, I found Sashoto Dotto. Is this deforestation event can affect the bee venom production?
Bee venom production, deforestation event. I'm sorry, if I understand correctly, so the question is...
Actually, the question is that due to heavy deforestation in these recent times, how this event can affect the quality of honey production, the first question. And is it also that have any neuroethological changes? And the next question is this deforestation event can also affect the bee venom production?
Okay, so the first part of the question is deforestation event on honey production. The second question is deforestation on neuroethology. And then the third part is deforestation effects on venom production, right? Great questions. So to answer the first part, deforestation event can definitely indirectly affect honey production. It also depends on what is flowering in that forest. So forests provide natural habitat for a lot of wild bees, not just honeybees, but also a lot of native bees and other wild bee populations. So if you are causing deforestation, you can, of course, you can destroy the natural habitat for a lot of bees. Deforestation can also reduce the availability of forage for bees, and that in turn can have an impact on honey production because they are not being able to bring back enough nectar to be able to produce enough honey. The second effect on neuroethology, so to understand if it has an impact on neuroethology, we need to be very specific on that stressor, not like an overarching, for example, is it this one particular pesticide? Is it the lack of one particular nutrient? So there are like these individual things that we need to parse out to be able to understand because it is complex. Like me, there are a lot of labs, in fact, a lot of labs who have been established for decades just studying bee neuroethology. We only look at one factor at a time because it is very hard to be able to tell that, what makes a bee behave the way they behave. It's very complex. And moving to the third part, if deforestation event, what was the third question? If it can affect what?
The effect of deforestation on honeybee venom production.
Right. So deforestation has nothing to do with honeybee venom. It is a very natural protective adaptive mechanism in the worker bees that actually the queen has a stinger too, but they have a venom gland with a stinger attached that they can use for in terms of emergency or protection or attack. There are, as far as I'm aware, there are two companies, one based in the US and one based in Europe, who commercially produce honeybee venom for people who are allergic to bee venom, for allergy immune therapies. And other than that, there are like small scale businesses who rear honeybees so that they can do venom therapy, like, for curing body aches or this and that. So there are some other smaller companies which do that. But unfortunately, deforestation has no direct role on how much venom a bee can produce.
Okay, ma'am. Thank you. Okay, so the next question is from Ankush Singh. I think you have already answered, but he's asking whether pesticides can impact on their productivity for honey and what are the measures for reducing this ill impact.
Sorry, could you repeat that? Your voice is breaking.
Okay, yeah. He is asking whether pesticides can have any impact on the productivity for honey and what are the measures for reducing this ill impact.
Okay. Thank you, Soumya. Thank you, Ankush, for this great question. So to answer your question, pesticides can impact honey production indirectly. They can reduce the population size of the colony. They can cause mortality in the honeybee foragers. So not enough bees bringing back enough nectar to produce honey. So that could be one way that pesticides can impact honey production. And by measures for reducing this ill impact, do you mean that how we can minimize pesticide exposures in bees? Then we can do it in a lot of different ways. As I mentioned before, we can be cognizant of the label directions, understand what are the different residual toxicities, how much of the dose to apply, follow the label instructions, avoid spraying during bloom period when bees are there in the landscape, avoid direct and indirect sprays on honeybee nests and colonies. We can also work with growers, like beekeepers and growers can work together in formulating strategies so that the bees are not directly or indirectly exposed to pesticides, but the growers are also able to protect their crops at that point of time.
Thank you, ma'am.
There is another question from Rajat Pal. Is temperature have any effect on pollination?
Absolutely. Excellent question. Temperature will affect if bees are flying in or out. If I just compare the Pacific Northwest with the Southeast region, the Pacific Northwest is much colder than the Southeast. So we have a smaller, I would say, span of time in the year where we can work in the Pacific Northwest compared to the Southeast. It's just that I have been working with Fahrenheit for so long, I'm now trying to think of it from Celsius perspective. It is crazy that we measure in Fahrenheit here. So if it's about like 50 to 60 degrees Fahrenheit, I would say about 12 to 14 degrees Celsius. It is too cold for the bees to fly out of their colony. So if it is too cold in the morning, if it is very dewy, very wet, the bees are not going to fly out. They are only going to fly out when it's warm enough, it's not very windy for them to be able to forage and pollinate. So yes, great question. Temperature definitely has an impact on how bees can pollinate.
Shomo asked the question from Dhrubajyoti.
Okay, sir. So he's asking, is there any substituent of phytosterol when nutrition is provided artificially? Yeah, that's the question.
Very interesting question. To answer that simply, there is no substitute for phytosterols. If you are providing an artificial diet, you need to provide synthetic phytosterols then. And I work with a lot of these artificial diets where I have to custom synthesize these phytosterols, and I label them either with a 13C isotope or with a 15N nitrogen. So I label these phytosterols and I provide it to the bees through their diet to see how the phytosterol assimilates over a course of time and what the temporal pattern is. Unfortunately, there is no substitute. A phytosterol is a phytosterol. So instead of trying to provide it synthetically, our best source is, as I mentioned, get it from natural sources. Either use like a vegetable oil supplement which has enough phytosterols that bees need, or trap for pollen during times in the year when there is plenty of pollen in the landscape and use it in your artificial diets in times of pollen dearth. So unfortunately, there is no substitute for any of these micronutrients other than adding it either synthetically or naturally.
Another question from Basu Dandapat. Is it possible to make arrangement to make bees to pollinate in different areas or place with a different range of temperature?
So if I answer that question correctly, that with different temperature ranges, can bees pollinate differently? Yes. So as I mentioned before, if the temperature is not conducive to the hive, the bees will not fly out of the colony to pollinate. So sometimes, let me give you this example. California, the almonds, they are blooming sometime around the end of January to the end of February or beginning of March, maximum. It's a very short bloom period, but an intense bloom period for a month. So when bees are being brought to California, it's January. It's very, very cold. So in California, it is still a little warmer in the Central Valley compared to all of the other regions that bees are being brought from. If it is cold in the morning, the bees are not going to pollinate. Only when it warms up during the afternoon, the bees are out pollinating the almonds. In Oregon, for example, the bees are then moved to pollinate blackberries or blueberries. Same, depending on the weather. If it is too cold, they are not going to fly out. If it is warm enough, they will pollinate that crop. So suppose if you have blueberries growing in a cold region and you have blueberries growing in a warm region and you place a bee colony in both, the blueberries growing in the warm region will have more bees flying there and pollinating compared to blueberries in the cold region unless it is warm enough for them to fly. I hope that answers your question, or I hope I got your question correct.
Yes. I think no question is left. Please, Somo, check once no question is left in YouTube and also our Google Meet chat box.
No, sir. I think this was it. This was the last question. And yeah, there is no question from YouTube or...
So from my side, a big thank and to Dr. Priyadarshini Chakraborty Basu for her excellent and very informative lecture. That is very much helpful for our student and also for us. So one thing that during discussion of the first lecture, I think we have missed one question. If Dr. Sayan Chakraborty is present, I think he is present. Then can I ask the question?
Yes, please, go ahead.
The question asked by from our one of our colleague from Professor Jayati Ghosh, how we can balance wound healing and tumorigenesis by Agrin. Though there was some similar type of the question, but how we can balance wound healing and tumorigenesis by Agrin?
Well, in cancer studies, we need to find ways to reduce Agrin expression, whereas in in the wound healing studies, we need to optimize ways of sustained application of recombinant Agrin proteins. So it's a balance of one reducing on the cancer and the other one is obtaining this optimal levels of the recombinant versions of Agrin. So that's how we need to balance out. And the fact is, in cancer and wound healing, they would be mutually exclusive to each other because in one case we are decreasing, and in the other case we are holding on by ectopic expression. that's by application in the form of ointments or something similar formulations.
Okay, thank you very much, Dr. Sayan Chakraborty. Meantime, another question asked by Pooja Upadhyay and also answered by our Madam, Dr. Priyadarshini Chakraborty Basu in the chat box. Then
exactly.
Please, Somo Mishra, please, you can ahead.
Okay, so I guess ma'am has already answered the question in the chat section. So I don't think so. I have to ask the question again, but I will read out the question for everyone. Okay, she asked which type of plants or crops enrich the bee nutrition in our country, which can be grown by the beekeepers. And ma'am has answered it. Should I read it out or is it okay? Okay, I'll read it out. Okay, ma'am, that's great question, Pooja. We are still trying to build the database, and the plants that I'm working with on in the USA are quite different from the plants in India, but a lot of them may also be common. Please always check your local planting regulation. Mustards and canola are good. Okay, this is the answer which ma'am answered to Pooja. I hope she's satisfied with this answer. Okay. Thank you so much, ma'am. I guess this was the last question, and yeah, there is no more question. Thank you so much. It is one of the most interesting webinar I have attended so far. Thank you so much, sir and ma'am.
No problem.
And
yes. I really enjoyed and learned a lot about bee.
Excellent, excellent.
Yeah, exactly.
That was an excellent.
We are very much fortunate. We are very much fortunate that both of the lecture from different area of the life science. So we are very much enriched and very much enriched. So again, thanks both the speaker, and I would like to hand over the next portion to Dr. Somaditya.
Thank you, Soumya. Thank you, Dr. Guria, for nicely conducting the interactive session. So thank you again, Dr. Chakraborty Basu and Dr. Sayan Chakraborty, for your precious time and for accepting our request and presenting this webinar. So we are at the final stage of this webinar, and the vote of thanks will be given by Dr. Jayati Ghosh, one of our colleagues, and she is actually associate professor at our department and the joint convener of this particular webinar. So Dr. Jayati Ghosh, with the vote of thanks, please.
Thank you, Somaditya. Am I audible?
Yes,
yes, madam. Yes.
Good evening, everyone. It gives me immense pleasure to propose the vote of thanks for today's webinar. And I, on behalf of the organizing committee, convey deep regards and hearty thanks to our eminent speakers who Am I audible?
Yes, madam.
Yes, yes, now you are audible. There were network issues.
So thank you, Somaditya. Good evening, everyone. And it gives me immense pleasure to propose the vote of thanks for today's webinar. I, on behalf of the organizing committee, convey deep regards and hearty thanks to our eminent speakers who spared time from their busy schedule to grace the occasion. And I would like to extend our deep sense of appreciation for Dr. Sayan Chakraborty for his explanation and enriching talks on mechanobiology of Agrin in cancer and tissue healing. It was really wonderful hearing from you about this novel area of research. And further, I would like to thank Dr. Priyadarshini Chakraborty Basu for making an excellent presentation on transdisciplinary approaches for improving pollinator health and making this webinar a very interesting and meaningful and highly interactive one. Thank you so much. And on behalf of the Department of Zoology, I express my deepest gratitude to our principal sir, Dr. Samar Chattopadhyay, for his encouragement and enormous cooperation in all our endeavors. We are thankful to Dr. Abhijit De, IQSC Coordinator, and Dr. Arun Kumar Roy, Teachers Council Secretary of our college, for being with us this evening. And I would like to thank our beloved head of the Department of Zoology, Dr. Shumona Saha, for her constant help and guidance that radiated a source of energy within us. And my deep sense of appreciation goes to the dedicated organizing secretaries, Dr. Srikant Guria and Dr. Shomaditya De, as well as a team of motivated students for working very hard to make this webinar successful. And I must also acknowledge the technical assistance and help provided by Mr. Supriyo Dash, Skyline Web and Application. And finally, I would like to thank all the participants, our colleagues from different colleges and universities, beloved students, for being present here and making the webinar successful. And now I formally announce the end of the webinar. And please submit your valuable feedback using the provided link within 24 hours. And certificates will be mailed to your registered email IDs after receiving the feedback. Thank you all very much once again, and good night for today.
Thank you. It's been a pleasure. Yeah, thank you.
Thank you so much for having us.
Thank you.
Thank you, ma'am. Thank you, sir.
Thank you. See you.
Thank you, Priyadarshini.